Taking the first steps in this journey, we will start by questions which may have troubled many of those concerning themselves with sports. The well-known nature versus nurture debate. How confined is our ability to grow and develop by our genetic material? What determines one's athletic potential? We will start then from the nucleus of our cells and the genetics, which will largely, but not absolutely, determine our potential. The human is a multicellular organism, made up by around 37 trillion cells of around 200 different types according to their task to bear. Let's take a look at what does a eukaryotic cell, thus the cells of all multicellular and many unicellular organisms, look like. In the center, there is the nucleolus, the nucleus surrounding it, containing our genetic material, and the nuclear envelope, an outer porous membrane enclosing the nucleus. The endoplasmatic reticulum is a folded, membrane-like system surrounding the core, which is responsible for the synthesis, storage, and transport of substances inside or outside the cell. Then, in a similar system, there is a Golgi apparatus or Golgi complex. This is where proteins are prepared and occasionally processed to be secreted outside the cell. Another organelle of the cell is the mitochondrion. The mitochondria are the energy factories of the cell, where nutrients will burn using oxygen to produce energy. The ribosomes, where proteins are synthesized, the lysosomes and peroxisomes, which are demolition centers, where cellular components are broken down, all bound together by the cytosol, which is surrounded and held by the outer layer of the cell, the plasma membrane. As mentioned earlier, our genetic material is stored in the core of our cells. The nucleus contains the deoxyribonucleic acid, well known as DNA, and the ribonucleic acid, the RNA. DNA and RNA are the biomolecules of heredity, because they store and transmit genetic information which dictates how an organism is structured and how it functions. DNA stores, while RNA transmits, where much of this genetic information ends up in protein synthesis. The sum of all genetic material is called genome. The genome of eukaryotes is organized in multiple chromosomes, each containing a duplex DNA. Human diploid cells, thus cells having two complete sets of chromosomes, contain 46 chromosomes in the nucleus in 23 pairs. Our DNA in each chromosome consists of 4 bases. These 4 bases, the deoxyribonucleotides, are adenine guanine, named as a group the purines, and thymine cytosine, named as a group the pyrimidines. They are connected, forming a chain. The sequence of the deoxyribonucleotides composing a DNA molecule is its primary structure. The most common secondary structure of DNA is a double helix. The double helix has 2 strands in opposite directions, attracted to each other by hydrogen bonds between the base pairs adenine-thymine and guanine-cytosine. The building blocks of RNA are the ribonucleotides. Just as in our DNA, differing in the aspect of not containing thymine but uracil. In order to survive and grow further, an organism needs to produce new cells. Most of our cells do so via a process called mitosis, through which the cell is divided in 2 cells, each equipped with an exact copy of the parent cell. Therefore, cells need to duplicate their DNA. This process is termed DNA replication. A protein named helicase will act to unfold the 2 strands of the helix, while DNA polymerase will add the deoxyribonucleotides to the new forming strand. DNA replication occurs only once in the lifetime of a cell, and this is how our genetic information is kept in our cells. But how is it transmitted and how does it affect our growth and potential in all its aspects? The genetic information flows from DNA to RNA. This process is termed transcription. The synthesis of RNA uses DNA as a template. The 2 DNA strands need to unfold locally at the site where transcription is to begin, with ribonucleotides being added one after another opposite the DNA bases, leading to formation of an RNA molecule that is called a transcript. Transcription is a never-stopping process in the cell. Many of the RNA molecules produced through transcription carry the information to build proteins. However, RNA needs to undergo extensive processing inside the nucleus before it becomes ready to transmit this information. Processing involves mainly the excision of segments from the interior of the so-called primary transcript and the splicing of the segments that are left. We call the segments that are excised introns and those that are left exons. Exons form the messenger RNA, mRNA, which contains the main information on the amino acid sequence in a protein. How does the 4-letter alphabet of RNA bases dictate 20 amino acids of proteins? 20 amino acids needed for a protein. There cannot be a 2 base to 1 amino acid matching because the number of possible permutations of 2 items out of pool of 4 is 16, 4 squared, which is insufficient. Therefore, 3 bases need to correspond to 1 amino acid, even though the resulting number of possible permutations, 64, 4 cubed, is much larger than necessary. The triplets of bases are called codons, and all possible triplets of RNA bases and the amino acids to which they are translated create the genetic code. The term gene refers to every part, region of DNA that is transcripted into functional RNA, that is, RNA serving a biological function. The number of genes in an organism relates to its complexity. Humans have 39,000 and they are divided into 2 classes. First, genes containing information for protein synthesis, 21,000, and second, genes containing information only for RNA synthesis, 18,000. The whole process above explained of the flow of genetic information contained in a gene to synthesize RNA or protein is called gene expression. When a gene is expressed, we refer to RNA and protein as gene products. The gene products will determine how we function under certain conditions and therefore how we grow and adapt in our environment and life circumstances or needs, since the gene products, thus RNA and protein, are the major contributing factor to all our organisms' function to feel, act and synthesize so that we adapt, grow and survive. Although the overwhelming majority of the genome is identical across all humans of the same sex, approximately 0.1% varies. Differences in genotype, thus the base sequence of specific genes or other functional elements in the genome, may be responsible for differences in phenotype, that is, visible or measurable traits such as colors, characteristics, cardiac output, hemoglobin concentration, and many more. As a matter of fact, that is a major factor that makes the potential to achieve high performance in a given sport differ among athletes. We don't possess the same genome, and although this is not something we can directly examine, it is visible in our measurable variables affecting performance and would depict the phenotype of an individual at a particular moment. Research has brought to light that there are genes associated with athletic performance. The ACE gene, a polymorphism of the angiotensin ID converting enzyme, is such an example. The I allele of this particular gene has been associated with endurance-oriented events, while the D allele is associated to strength and power-oriented events. Our genome would define our potential in sports, but as direct and predefined as it sounds, the way we grow and adapt seems more dynamic. Exercise, with all factors affecting it, can affect all stages of gene expression, meaning that our genome would dictate our top, but how close we can get is dependent to many more factors, which we can more or less control or manipulate, those being our motivation and will, the social and parental environment, the society's support, our diet and lifestyle, the environmental conditions in which we live and train, the ability of the coach to guide the athlete in the best possible way. It seems a lot to consider before accepting our genetic karma, rather than being driven to discover it. Moreover, researchers have discovered that certain phenotypic characteristics can be passed from one generation to the next without being encoded in the base sequence of genes. This discovery led to the development of the field of epigenetics. Evidence is supporting that both acute and chronic exercise can cause epigenetic changes. Epigenetic changes leave their print on the phenotype by affecting transcription rate. In simple words, genes can be repressed by DNA methylation or inducted by histone acetylation. And most importantly, some epigenetic marks can be transmitted to the next generation as the human is selecting information about the world while still an embryo. Being conscious about what is our genome and how it will affect our adaptations and growth as humans and athletes, we understand that our possible best would be dictated by it, but how close we get to reaching our pinnacle of performance as humans and athletes is also affected by our choices and lifestyle. This as a fact illuminates the role of sports in our society. As an important part of our culture, sports is and will be a stimulus, a cause, and a need for us humans to strive to be the best possible version of ourselves in each and every aspect of our being, creating a better world for the present and the generations to come. Be yourself, best yourself.